Hello and welcome to this lecture on the basic topography of the central nervous system. My name's Steve Jakes, and you will have encountered me in a variety of contexts, mostly teaching anatomy, but this is the area that I'm really interested in, and that's neuroanatomy. So you can contact me on these various methods, email, rules permitting. You can come and see me in the Morris Shock building. You can view my videos on my YouTube channel, and you can also contact me through Twitter. But without further ado, let's take a look at what we're going to look at in this lecture. And the basic remit of this lecture is largely to recap what we've already looked at in previous semesters. So some of this, in fact most of this, should be quite familiar to you, but we are going to start gradually introducing some new concepts. So we're going to look at the basic components of the central nervous system. We're going to consider the differences between grey matter and white matter. We're going to take a look at the spinal cord and use the spinal cord really to try to understand what the basic plan of the central nervous system is. We're going to revise some simple features of the brain, and we're going to introduce some new ones. And finally, we're going to take a look at how cerebrospinal fluid is made and how it eventually leaves the CSF system. So let's kick off by recapping what's already been covered and where it has been covered in the course so far. Within body logistics, we've looked at simple areas like information flow in the nervous system, the basic bits and pieces of the central nervous system particularly. We have looked at CNS versus PNS structures, and you've already had a short introduction to the basic terminology regarding nervous system structures. Head and neck has covered the majority of your neuroanatomy to date, covering basic fundamental properties of nervous systems, looked at the superficial features of the brain, a bit more terminology, but you also looked at some of the deep features of the brain in head and neck, as well as the meninges and their clinical relevance. Not to forget also cranial nerves. And then remember, in MSK, you did quite a bit of neuroanatomy actually. So you looked at peripheral nerves, dermatomes, myotomes, and the importance of the chord requiner. And a large part of MSK was in fact dedicated to nerve lesions, those specifically regarding the peripheral nerves. Now we're going to be building on most of these things. But a large part of our remit for this unit is going to be using this knowledge to localize lesions within the central nervous system. So you've done peripheral nerve lesions, but a lot of nervous system is going to be looking at central nervous system lesions. Now let's revise the very basic components of the CNS. You should recall that the CNS, the central nervous system, is made up of the cerebral hemispheres, the brainstem and the cerebellum, and the spinal cord, whereas the peripheral nervous system, the PNS, is formed from the dorsal and ventral roots that plug into the spinal cord, the spinal nerves, and the peripheral nerves that are ultimately formed as branches of the spinal nerves after passing through various plexi. Now on the left hand side here is a specimen which always fascinated me as a child. This was in the Science Museum in London, and I don't think it's there anymore. But someone has painstakingly removed the brain and the spinal cord to produce an incredible potted specimen showing the central nervous system. And what you've got to realize in preparing this specimen, a huge amount of bone and soft tissue had to be removed to expose these structures, just reminding us how delicate this tissue is. So the brain and the spinal cord is incredibly delicate, and that is why it is enclosed within the skull and the vertebral column. Furthermore, you can also see connecting to the spinal cord these various elements of the peripheral nervous system, and we can see here, for example, some of the dorsal root or sensory ganglia connecting to the cervical spinal cord at the level of the brachial plexus and connecting to the lumbosacral cord at the level of the lumbosacral plexus. If we look at a cross section here through the spine, we can see the spinal cord sitting centrally, and we can appreciate that we have these peripheral nervous system structures plugging in to the lateral aspects of the cord. And we're going to have a look in much greater detail at this during much of the rest of this unit. Finally, let's have a look at once again the brain and cord, but in the context of the entire head. And we can see here is the cerebral hemisphere cut down the midline in the sagittal plane. We can see one of these dural folds. This is the falx cerebri here, separating the two cerebral hemispheres. We can see the cerebellum at the back, and we can see the brain stem going down into the spinal cord within the vertebral canal. So it's important that you have an appreciation of these structures, where they sit and the basic properties that they have. Now, two terms are used when we talk about the central nervous system. These are histological terms, but we can also actually see these macroscopically as well with the naked eye. These terms are grey and white matter. Now, the term matter was adopted by ancient anatomists who didn't really understand what the different parts of the body were made out of at the histological level. So they said, well, there's this stuff that looks kind of grey, and there's this stuff that looks kind of white. So we'll call it grey and white matter. And these terms have persisted to the present day. Now, grey matter is primarily made up of the cell bodies and dendrites of neurons. So the grey matter is where the cell bodies are. The grey matter is where there is a huge amount of metabolic activity taking place. And that means that the grey matter needs to be highly vascularized. Highly vascularized to provide the oxygen and nutrients to feed the synapses which are performing the computations within the grey matter. Now, white matter is made up of axons. And of course, axons rarely are found alone. They often have supporting cells with them. So grey matter is made primarily of cell bodies and dendrites. White matter is made up mostly of axons. Now, that's not to say that grey matter does not contain axons, because it does. Axons join up areas of grey matter. If there weren't axons going into and coming out of areas of grey matter, they wouldn't be able to communicate and the nervous system wouldn't work. So there are axons within grey matter. However, they are far less prevalent than the cell bodies, dendrites and synapses which are found there. Now, why is white matter white and grey matter grey? Well, this is primarily due to the presence of myelin within white matter. Now, whenever you have a mixture, an emulsion of a fat and an aqueous phase, you tend to get a white coloration. Think about mayonnaise, for example. And so all that myelin is really is extremely sophisticated mayonnaise, giving white matter that white colour. Grey matter has less myelin and so appears greyer, less white. And furthermore, the presence of all these blood vessels also alters its coloration. Now, some of these terms have equivalents in the peripheral nervous system. So the peripheral nervous system equivalent of grey matter is a ganglion. So the term ganglion is used to refer to a collection of cell bodies in the peripheral nervous system. However, beware of the term basal ganglia, which once again is one of these archaic, old-fashioned terms introduced before the terminology was standardized. And the basal ganglia are central nervous system structures. They are not elements of the peripheral nervous system. And the peripheral nervous system equivalent of white matter is peripheral nerve. So, for example, the ulnar nerve could be thought of as the equivalent of an area of white matter within the central nervous system. These images on the right show a variety of relevant cells. Here on the top in pink we can see a motor neuron from the spinal cord. We can see the large cell body here, its axon emanating towards the right hand side of the image, and all these dendrites converging down onto the cell body. In the middle we've got a beautiful fluorescence micrograph of an oligodendrocyte, and in green in the background we can see all the axons that this oligodendrocyte is myelinating. So one oligodendrocyte, as you should recall from body logistics, can myelinate many axons, and these are lovely cells to look at. And finally at the bottom we're looking here at sensory neurons, pseudo-unipolar neurons within the dorsal root ganglia, and we can see here is the cell body, and here is that single axon coming out, which we're going to learn much more about in later lectures about what this axon and its branches do. So that's grey and white matter. Now let's look at some more CNS terminology. So we have this term nucleus, and a nucleus is a grey matter structure. A nucleus can be defined as a collection of functionally related cell bodies. And we're going to learn about many nuclei within the central nervous system. But the key thing for you to appreciate at the moment is that a nucleus is made up of a collection of functionally related cell bodies. The term cortex once again refers to grey matter, and the cortex is a folded sheet of cell bodies found on the surface of a brain structure, typically the cerebral or cerebellar hemispheres. Now the cortex is folded and contains many cell bodies, typically something between 1 and 5 millimeters thick. In fact, the word cortex comes from a word referring to the bark of a tree, and that reminds us that it sits on the outside, just like the cortex of the kidney, for example. Finally, a fibre, this being a white matter term, is a term relating to an axon with its supporting cells. So for example, a central nervous system axon surrounded by myelin sheaths made by oligodendrocytes can be considered to be a fibre. And we frequently use the term fibre synonymously with axon, although axon, strictly speaking, refers just to the axon and not to the supporting cells. Now within the brain and spinal cord there are 3 particular types of fibres present. We have association fibres, which are seen within the same hemisphere of the brain and which are seen to connect cortical regions together. So there are association fibres connecting, say, the temporal and parietal lobes together on the same side of the brain. We have commissural fibres which cross the midline in order to connect the left and right hemispheres of the brain or spinal cord. And the big commissure in the brain is the corpus callosum, which is a large structure connecting the left and right cerebral hemispheres. Finally, projection fibres are those which connect the cerebral hemispheres with the spinal cord and brainstem, or vice versa. And a large part of the first half of this unit is going to be learning about the pathways of these various projection fibres as they descend to form the motor systems, or as they ascend to form the sensory systems.